Good morning, everyone, and welcome to Lunch with a Scientist. We hope you had a nice little break for the holiday. And so this is your chance to get to know another awesome professional scientist. Today, we're going to welcome Nate Olson, who is going to tell you a little bit about his work. And so Nate earned a PhD in Bioinformatics from the University of Maryland in 2018. He's worked as a research biologist at the National Institute of Standards and Technology for 10 years on various projects ranging from evaluating methods for biothreat detection to genomics and metagenomics. So he's currently part of the NIST Genome in a Bottle team, working on standards for whole human genome sequencing. And if that seemed like a little bit of a mouthful, we're going to learn all about what that means today and hear more about his work. So I'm very pleased to welcome Dr. Nate Olson. Hello, Nate.
Well, thank you for the introduction.
You're very welcome. And where are we talking to you from today?
My basement at my house outside Washington, D.C.
Great. So a little bit after lunch for you, just early afternoon.
Yes.
All right. Well, we're super excited for your talk. So without further ado, I will let you get to it.
Yeah, thank you. And I'm excited to talk to you about metrology and genome sequencing in my work at NIST. So just make sure everyone's clear. We're talking about metrology here, measurement science, and not meteorology. While the weather is very interesting, I'm more interested in measurement science and how we measure things and how we do it as accurately as possible. So I'm going to give you some brief background because most people are unfamiliar with metrology and NIST. So in 1875, a number of countries got together and signed the Treaty of the Meter. And what this did was it defined a common set of units for measurement. So meter, kilogram, second. These are used in commerce so that if you buy a gallon of milk or a liter of milk in the United States, you have the same volume as it is in Canada or Europe or other countries. Shown here is a picture. In the center is a picture of BIPM. That's a International Metrology Institute. It's an intergovernmental scientific organization that's located outside Paris, France, where the treaty is signed, and it's still the International Center of Metrology. And that's where the SI, Scientific System International, or scientific units are defined, such as the mole, candela, kilogram, etc. And at NIST, National Institute of Standards and Technology, which is part of the United States Department of Commerce, where I work, is the United States National Measurement Institute. And so what we do here is make sure that measurements made in the US are consistent with an international system, the SI, that facilitates commerce both within the US and international. So it plays an important role in all our lives, although most of us never heard of the organization. I hadn't before I started working here, working at NIST. And so one way that NIST plays a role in our lives is through enabling advances in technology. Without a precisely measured second, the SI for time, we wouldn't be able to have GPS or cell phone technologies. A lot of great work has been done at NIST for developing atomic clocks and measuring seconds very precisely, and this enables high accuracy GPS measurements. And recently there's been some fun, interesting advances in metrology, redefining the kilogram. So initially, all the standards, there were physical objects. Shown here is the last physical object for a standard, which was the grand K for the kilogram. It was a platinum cylinder. Whereas, and for example, with the meter, it was a platinum rod. However, these physical standards can change over time. And so they've been slowly redefining all the scientific units to be physical constants of nature, where second is the speed of light traveled. Light traveled over the time that it takes light to travel a meter, a specific type of light. And they redefined the kilogram so that it's based on, it's a unit of measure for both meter related to the meter and the second by using Planck's constant. And so some amazing physicists and chemists use a thing called the Watt balance to redefine Planck's constant and remove, so we no longer need the physical artifact to define the kilogram. And this works through this pyramid of standards, and we have traceability of measurements to the SI units. So you have kind of process measurements. If you think of going back to the gallon of milk example, that's the measurements that are used when they're bottling the milk. And you can have working standards or standards that are used by the people who produce the measurements that produce the instruments making these initial measurements. And then secondary standards are used to calibrate, ensure that the working standards are correct, and then primary standards. And those are housed by the different NMIs, or National Metrology Institutes, such as NIST for the United States. And those are all under the BIPM, or International Metrology Institute, where was responsible for defining the scientific unit. So it's a valuable and somewhat complex hierarchy, but it's important to understand the traceability and being able to have the SI units defined, enabling technology, but also as working exam, but being able to use them in through your work. However, I do biology, and we're kind of like in the wild west of metrology, where we aren't able to trace our measurements back to any scientific, to an SI. For example, with DNA sequencing, we can use the kilogram to measure the mass of nucleotide, the subcomponent that makes up DNA, the bases that are strung together, and we can use the mole as a measure for the number of nucleotides, for the different nucleotides that are in a DNA sequence. But it's that ordering of the nucleotides that make DNA, that enables DNA to do what it does and provide a blueprint for life. And so we have to this is both a challenge and an opportunity for our work, and what makes it very interesting, challenging and interesting. So I work on a project called Genome in a Bottle, and what we do is we develop standards for human genome sequencing, and we use a consortium, which is a collaboration of government agencies, private industry, and academia to develop standards that enable people to evaluate and validate their genome sequencing methods. And we develop standards, which is a genome in a bottle. So we have DNA from different individuals. It's available for purchase that people can take, and they'll sequence, and they will compare their sequencing results to our characterization of the material. And so what they're doing for their sequencing results is they're actually comparing variant calls. So in the process, we start with our genome in a bottle, DNA. We pass it to a DNA sequencing instrument, and then do some analysis or bioinformatics. And for this, we take the individual sequencing data that comes from the instrument, and we compare it to a reference genome. This reference genome that's developed by the folks at the National Institute of Health, but by comparing the sequencing results to this reference genome, we are able to identify variants or places in the genome where our individual differs from the human reference genome. And so then what we do is we allow people to evaluate and validate how well they're able to identify and call those differences. But how do we characterize our material? How do we identify these variants so that we can identify them in a way with high confidence? And we do this in collaboration with a number of people. We have a recent publication that's undergoing peer review right now. And what we do for this process is for our characterizations, we take sequencing data following this diagram here. We use sequencing data from multiple technologies and multiple data analysis methods, and we use a sophisticated machine learning algorithm to combine the results. So we have our characterizations, and we use a combination of expert-defined heuristics and parameters and machine learning methods to generate high confidence characterizations. And these are where we know that our genome is different from the reference genome and is the same. And this allows our users and stakeholders to evaluate their measurement results. But how do we know that our characterizations are correct? And this is where we have to take our own unique take on metrology. And we do this through crowdsourcing and defining traditional metrology terms and using a functional definition. So in metrology, validation is determining whether a measurement process or value is fit for purpose. And here we're defining fit for purpose is that when a user of our reference material compares their sequencing results and their variant calls to our characterizations, any differences or discrepancies are primarily errors in their sequencing data or variant calling methods and not our characterizations. And so what we start here with our genome in a bottle of genomes or DNA, and we collect measurement data using multiple sequencing technologies and analysis methods, and we process it through our integration pipeline that I just described. And we have our candidate benchmarking set. And then we do a crowdsourced benchmark set evaluation, where we have our collaborators provide their variant call sets, and we compare their variant call results to our characterizations using our benchmarking pipeline. And any differences that are identified, we manually curate them. And so by doing that, we look at how well this individual sequence data supports their variant call and our variant call. And if most of the differences after manual curation, if the sequencing data supports our variant calls more often than it supports their variant calls, then we say that our characterizations are fit for purpose, and we release our benchmark set. The publication that I just showed describes the fourth version of our characterization. So this is an iterative process. And then our stakeholders users of our reference materials can use the genomic DNA and our benchmarking methods to evaluate their methods using our benchmarking pipeline, because it's not easy to compare the variant calling results. And so we developed the data analysis or bioinformatic pipeline to do this. And what's great about our work is it's an iterative process. And so while we can have our users will provide new variant call sets, and it allows for us, if let's say a new variant call set identifies some errors in our benchmark set, we can go back and then incorporate their variant calling methods into our integration pipeline and generate a new benchmark set. And so our benchmark set and characterizations allow people to refine and revise their genome sequencing and variant calling methods. But their genome sequencing and variant calling can in turn also allows us to improve our characterizations as well. So it's a positive feedback loop. And a great example and application of our benchmark sets was a recent truth challenge that was hosted by the FDA on their Precision FDA platform. And what we did was we provided our sequence data for our 3 of the genome and about genomes, and asked participants to submit variant call sets for these genomes. And we provide sequencing data from multiple technologies. We were interested in seeing how well they performed in difficult, more challenging regions of the genome, because the sequencing data, it's not one whole, you don't get the whole genome at once, you get it in pieces. And so a lot of the analysis is taking those pieces or short DNA sequences and figuring out where they go in the genome and identifying differences. And so there's parts of the genome where there's high similarity between multiple regions of the genome, such as segmental duplications, which is a part of the genomes where you have multiple copies of similar DNA through copies that made through evolutionary time, and low complexity regions where you have a lot of individual nucleotides in a row, nucleotides that A, G, C's, and T's. And so with this challenge, there were 20 participants, and they provided there were 64 submissions. And interesting, a lot of them are using some new machine learning based technologies where leveraging some of the same algorithms that are used for image recognition, differentiating cats and dogs in Google image search, etc. And by using multiple technologies and these new machine learning methods, they're able to identify variants with a really high accuracy. So here showing a couple of plots. On the x-axis here is the F1 score, which is a combination of the precision and recall. So precision is how often when they call a variant or identify a variant, how often it's correct. And recall is whether the variant, how many of the true variants they're able to identify. And so here we're showing is a histogram and showing performance for different parts of the genome. All our benchmark regions is all the genome that we've characterized. Difficult to map regions, which are a lot of the low complexity regions where you have strings of A's and T's, long strings of A's and T's, and then MHC is a major histocompatibility complex, which is part of the genome that's particularly difficult to sequence, but medically relevant because it plays a role in your immune system. And what we saw is that participants who use this PacBio, which makes a sequencing technology that has a longer sequencing reads about 10 to 20 kb, but a higher accuracy, and as well as using multiple technologies. In the brown and the orange performed the best. And so it's these new technologies, new submissions, these challenge submissions, challenge participants developed some interesting new variant calling methods that performed extremely well. And you can see here where I'm showing the individual submissions are connected by lines. You see this is participants that use these machine learning algorithms and multiple sequencing technologies were able to perform well because they are able to leverage, take advantage of the different strengths and weaknesses of the different variant calling methods. And this is, in one way, this is how Genome in a Bottle enables commerce, is that participants in the study were, there's a group from the Google health team using their new deep variant algorithm, which leverages the deep neural networks similar to the image recognition technology for high accurate variant calling. And also, and these are showing here is blog posts by 3 different technology companies. Google, as you are all probably familiar with, there's also Illumina, which makes a common, widely used short read sequencing technology where their sequencing reads are 100 to 300 base pairs in length, but much higher accuracy than the long read technologies. And then again, PacBio is another long read sequencing technology, but also has a unique method that allows for high accuracy. And so by hosting challenges and developing our reference materials, we provide a way for individual different technology developers to evaluate their methods, see how well they do in comparison to other technology, other companies, other developers, but also understand where they may not be doing as well and where they can work to revise and improve their methods. So I want to kind of wrapping things up a little bit. I do want to acknowledge that the work that I do is fun and exciting, and I think that I can't do it alone. There's a great team that I'm part of at NIST and the Genome in a Bottle Consortium, working with people at companies all over the US and the world, really makes it interesting and allows us to tackle challenging problems that we wouldn't be able to do. We wouldn't be able to do alone, but also through this work, we're able to enable commerce and help companies work on building better sequencing methods and variant calling algorithms without having to worry about figuring out how to know if they're getting the right answer or not. And so I want to thank all of you for your time, and I'm happy to answer any questions that any people who are watching may have.
That's great. Thank you so much, Nate. Really fascinating to learn more about a subject that's really important to us humans. So while we're waiting for the audience to type in any questions they may have, I wanted to kick off with just maybe some inspiration for the students who might be watching. So one of the things that I didn't read originally in your bio is that you started out your academic career in marine microbiology and then eventually found computer programming and metrology as your passion. Can you talk a little bit more about how you made the shift and how you knew what felt best to move forward in your academic career?
Yeah, so I started out before marine biology, I went to school briefly for engineering, and it wasn't a good fit for me. And I was a little unsure of what I wanted to do, so I took a few years off, and then I had a passion for marine biology, coral reefs. I really liked corals when I was younger, and so I went to Hawaii to study marine biology. Had a great time there, got to go scuba diving, study corals. I did a lot of work in the lab doing molecular biology, learning about symbionts, the bacteria that live inside corals that make coral reefs part of the amazing living organism and ecosystem that they are. And then I went on to get a master's in microbiology from University of New Hampshire, and I still studying corals, but I took a computer programming class. And all along the way, I also really enjoyed methods. Some of the most fun I had as an undergraduate was trying to figure out the best way to extract DNA from corals, which is really hard to do. They're slimy, and it inhibits some of the reactions. And so I was interested in methods and programming. I went to a conference and ran into someone from NIST and just kind of worked out that's where I got a job. And I think it took me a while to get to where I am, but I think it's important to find things that you're passionate about and let them kind of guide you. And I think along the way, feel free to take as much math and science classes as you enjoy, because I think no matter what you're going to end up doing, it'll be helpful, especially the math. So.
Right, to have a strong foundation no matter where you end up. Great, thank you so much for the advice. So we've got a question. What advances do you expect these new techniques in genome sequencing to bring about in healthcare and other applications?
That's a good question. I think there's been some real exciting breakthroughs and advances in sequencing technology. I think in around 2000, they finished sequencing the human genome, and they use a method called Sanger sequencing, and it was a worldwide effort to sequence one genome, and they never finished it. Few people actually know that the human genome has not been fully sequenced. There's a group now, the T2T consortium led by Adam Philippi at NIH and Karen Miga at UCSC. They're working on finishing genome with new sequencing technologies. There's the high accuracy short reads, Illumina, that I talked about, but also the longer sequencing methods, longer read, relatively high accuracy from PacBio that they're using to develop some high quality genome assemblies. And there's another sequencing technology, Oxford Nanopore, and they have a small sequencer that's about the size of a large thumb drive that reads the DNA through, and they're able to get real long reads. So Illumina reads are a couple hundred base pairs, and the Oxford Nanopore reads, they have some that are 1 million bases. And that really helps putting together a genome that's 1 trillion bases, 3 trillion bases. And so how this helps in healthcare is that by the better we're able to sequence and characterize the genome, the more the genome that's going to be accessible, and the more accurately we're going to be able to identify variants and call variants in places that we may have not even been able to characterize before and have unknown medical relevance. And as sequencing gets less expensive, it'll be in a more commonly done to really improve diagnostics and help people identify causes of disease in a rapid, accurate manner.
Mm-hmm. And when you're talking about variants, you talked about the Genome in a Bottle project and looking at someone's DNA in comparison, I guess, to the standard. How is the standard determined?
So we have standards for genome sequencing, and then there's the human reference genome. It's confusing. Even confused people that I work with outside our group with overuse of the word standards and reference. But there's the reference genome is kind of like the canonical reference. This is what people say, this is the human genome, where it's actually a composite of DNA that was from 7 individuals that was donated and used to make the first human reference genome.
So it sounds like that's what we're measuring against when we're looking at specific variants.
Mm-hmm.
Interesting. Okay. And so this kind of relates to this next question, which is coming in on the public front. How does this kind of work relate to these kits that people can buy at home, like 23andMe or other home options that are coming out?
So how our work relates to that is companies like 23andMe and others, they can and do use our standards to evaluate their sequencing and data analysis methods, so that our standards help improve the quality of the data and results that you get from these companies.
Mm-hmm. Interesting. Now, going back a little, just a fun question about your marine biology path. Mari wants to know, what's your favorite fish?
I'll like to say the humu humu nuku nuku apua'a. It's the Hawaii state fish.
Great. I love it. For those of you who may not know, I'm originally from Hawaii, and Nate went to school there, so we've got kind of an interesting connection. So I think we're going to wrap up here in a little bit, Nate, but I want to do one kind of final question for, again, our students who are watching. And you talked a little bit before about how you found your career path and how it felt okay for you to kind of try a lot of different things and then find what you loved in the end. For someone on the other end who really knows that they want to go into work with DNA sequencing or something very similar to what you do, what academic path would you recommend for them?
I think there's 2 ways to approach the work. One is from more of a biology, molecular biology perspective, and that's kind of often referred to as the wet lab. So going from the DNA, the cell culture, the biology, getting that into the sequencing instrument, and then taking that data from the instrument and doing the analyses. Both sides require having an understanding of the basic biology. And then if you want to do more of the wet lab, working in a lab and doing the pipetting, good understanding of chemistry, organic chemistry, really kind of being able to think at a molecular level, like what's going on inside the tube is very helpful in troubleshooting. Because most of the time when you're doing science, it's really just figuring out how to fix something that went wrong, whether it's in the lab or doing the programming. And then for the bioinformatics side, programming is very helpful. Some computer science, minimum, I think, math and statistics. I think I always wish that I took more statistics and math. It's really helpful and helps you understand how the algorithms work and why there's differences in analyzing data.
Mm-hmm. All right, well, there you have it. If you're interested in that career path, great advice from someone who's been in the field a long time. So, Nate, thank you so much for being here and presenting your powerful work. And I hope you have a great day.
Thank you, you too.